Good morning, everyone. My name is Ornit Nahman, and I'm a PhD student at the Shinor Lab. And I'm here to present the project I've been working on for the past months. Let's start. Okay, so short introduction. Spatial transcriptomics is a it's a relatively novel technique, and Dana kind of gave me a good intro with her slides to this. It's gained a lot of popularity, especially spot-based technologies such as Visium, and it just gives us a spatial context alongside with the gene expression. But spatial data has its own unique characteristics and challenges, you can say, and some of them are listed here. So we have mixing of cells of spots, which means, do I have a pointer or something? No, okay, we'll deal with it. So we usually have more than one spot in each location. Leakage of transcripts. So transcripts can diffuse to neighboring spots, and you can see the black line is the tissue boundary, and you have UMIs outside of it, and low depths per cell in a spot. So in comparison to single cell technologies, the coverage that we get per cell in a spot is much, much lower. Okay, so differentially expressed gene algorithms are in wide use also on spatial data, and most of them are the classic algorithms that have been developed previously, and they do not account for the new characteristics or properties of the spatial data. And we were trying to ask ourselves, are those the right tools for the task? So I don't know if it's clear from the image, but it's a person trying to eat ice cream with chopsticks. So the aim in this project were to build a framework for evaluating the differentially expressed genes performance on spatial data, to investigate to see if there are factors that can influence the ability to accurately recover the DAGs, and lastly, try to give best practices or see if we can even enhance the recovery based on our findings. So as per the first aim, I developed a simulation to help me. I had two goals. One is to curate realistic spatial data, and the second was to have a ground truth, which we already also mentioned earlier, that is a very problematic thing that we usually don't have anything to compare to. So we started with a single cell simulator, then we mixed the cells to spots. It will have a tissue structure. And on top of it, we added leakage and sparsity, so it downsampled the transcripts. So the first question we wanted to ask is, how do the common DAG algorithms perform on the simulated realistic data? And to do this, for reference, we use the single cell as a baseline. So you have here two evaluation matrices that we used, a sensitivity and the F1 score, which is more of a summary score that combines sensitivity but also gives us a little bit more of it. And in both cases, if you compare the simulated single cell to the realistic configuration, you can see that there is a dramatic loss in the scores. And this loss is also independent of the method that we use. So the variation between methods were quite small. So this was quite surprising. And with this, we were trying to understand what might be the factors that influence those scores that we received. To do this, because it's a simulation, so we can separate the different parts. So we try to separate the factors. And here we have, again, the F1 score, but this time, besides the single cell and the realistic configuration, we also have the mixing with leakage and mixing with downsampling. So we can try to see how each of them affects. And here again, we can see that there is pretty low, like the scores are still pretty poor for all of them. And even when we only add the mixing, we already see this notable decrease. So this made us conclude that it might be the mixing itself that is causing this effects that we see. Wait. Okay, what happened? Something is off. I can't go to the next slide. Help. Is it out of presentation mode or something?
I think so.
Yeah. Don't reduce this for my time. Okay, cool. Working again. Okay, so the obvious solution for the mixing or the heterogeneity in the spots is applying a deconvolution algorithm. So we tried to do this. And there's actually a relatively new DEG algorithm that has, as a first step, a deconvolution. Deconvolution is giving us basically the ratios of cells in each spot. And then this algorithm that is called Seaside is using this to detect cell type specific differentially expressed genes. So we try to use this on our data to see if it helps. And it's also important to mention that as part of this algorithm, it also filters a lot of the genes. So it doesn't evaluate marker genes and genes that have what it's called low confidence. Like if they're not in enough of the spots, then it doesn't give a prediction for them. So in order to keep the comparison fair, we also we took only one standard DEG algorithm, a very simple one, which is t-test, and we made sure that we apply them on the same subsets of genes. And we were, again, quite surprised to see that when we were doing this, when we were using the same subsets, we suddenly don't see much of an improvement for this algorithm. But it did give us an idea, thinking, okay, maybe we can try to think of another way to get a better confidence of our results. Maybe even without using deconvolution, maybe we can try to think of a smart subsets of genes that we'll have more confidence on. So we proposed a gene specificity scheme that is using, basically we're calculating gene specificity per cell type. And then if the specificity is high, it means that it should be more robust to the mixing. And as analogy, I have here a washing machine where all the clothes go together, so they're mixing up. And then in the laundry, when you dry it, you can see that there are red stains of some of the shirts. And it's the prime suspect is quite obvious because it's the property of only of one of the t-shirts. So that's the idea behind it. And here you can see the results. Okay, am I sticking too fast? So we have here the F1 score as a function of the specificity threshold. And when we increase the specificity threshold, it means the gene is more specific to the cell type. And you can see that, so we have a standard DEG again with our selected genes. For control, we also have the standard DEG with randomly selected genes, and we included Seaside. So each column here has the same number of genes tested. And you can see that when we select the genes as I proposed, then the increase in the F1 score is the most notable. So this have a downside because in this way we are not getting a prediction on all of the subsets of, like, on all the space of the genes. And on the bottom two graphs, you can see how many genes are lost in this process. So this is for the simulated data, but I also checked if you do something similar on a real single cell data, how many genes are retrieved. And even on the most strict threshold, it's still a quite useful number of genes. So to conclude, in this work, I built a framework with realistic spatial data and DEG ground truth. Then I tested standard DEG methods and showed that they recover the DEGs ineffectively on spatial data. I identified that spot heterogeneity is the primary factor that is contributing to the suboptimal results. And lastly, I suggested a gene selection scheme that can enhance the confidence of the DEGs that you're retrieving. So on these subsets, you know that the DEGs you're getting are, they're more likely to be true DAGs. And this method is this scheme is easy to implement and does not rely on the convolution to work. Okay, with this, I would like to thank you all for your time and attention, and to my amazing lab members, specifically my PI Shai, and the chief scientist team that I think is also here, came to cheer me up, the funding sources, and my dear family that stayed at home. That's all, and I'm glad to look at questions.
Thank you for your interesting presentation. I really like it. And if we have some questions, we have time for questions of the public. No?
There is one.
And in the chat also we have.
Yeah, a question in the chat is more like general.
Can you speak louder, please?
Okay. They're asking what is so Mia is asking, what's the measure of similarity or comparison between actual single cell data and simulated data?
Okay. So I think I checked some distributions of the mean and the variance, and also when I generated the leakage. And so I have measures. I didn't say. Okay, let me go back to this relevant slide. So I actually have 2 checkpoints along the way. So after the single cell simulator, I have quality control to make sure that the ground truth stays close to the simulated data. For the mixing, I'm using a real tissue to put the cells in a spot, and also the number of cell in a spot is it's a very long. It's something you can control. It's a parameter you can control, so it should cover a large realistic range, even besides, but also for the leakage, I use also parameters that were evaluated from real data, and for the sparsity, I again checked that the distributions are close to the ones that are in real tissue. So I kind of try to make sure along the way that I'm keeping it close to real data.
So just double check, you're doing a differential expression between 2 spots, right? Or groups of spots?
So I did this per cell type.
Per cell type, per spot, though?
The way I'm including all the spots that have the presence of the cell type. So it's not exactly per spot.
So you're basically trying to compare cell types across spots?
Yes.
Okay, thank you.
Hello. So this spatial transcriptomics data seems like it's very useful and can answer a lot of different questions that we weren't able to answer with gene expression. So I wanted to ask, how scalable are getting this spatial transcriptomics data compared to regular gene expression? With gene expression, you can do a microarray assay and measure over a variety of conditions for not a terrible amount of money. But so what's the scalability of this type of method?
What is? I'm not sure how to answer it. So can you repeat just the essence of your
Yeah, just in terms of how available this technology is to get the spatial transcriptomics data compared to normal gene expression. Can you get a similar amount of data for a similar price, or is it?
All right. I think it's very dependent on the methods, on the technology, and I specifically focused in my work on the one that is most popular and the cheapest, I think easiest to apply, which is spot-based or Visium. So I think it's relatively easy. I don't think it's still more expensive, as far as I know. And then there are a lot of others that the ones that really give you single cells have other pros and cons, and the pricing I'm not really too familiar with it, but I think it's getting there. It's like any technology; it always takes time to refine it and make it more available to everyone. So
Yes, thank you.